to speak about the special senses, sight, hearing. We need to first think about the idea of sensing versus sensation. So we want to start by defining these terms and then giving some examples. So what is sensing? Sensing is purely the physical detection of stimulus by a sensory receptor. Okay. Sensing does not infer or imply any conscious meaning given to those sensations. All right. So to say that I can perceive visual sensation or my sensory receptors in my eye can detect stimuli, but it's not until my brain, specifically the occipital lobe, right? The occipital part of my brain can add meaning to those sensory stimuli. All right. And that is sensation. So in some cases, sensing will automatically result in sensation, right? We're going to have the perception by a receptor, and then our brain is what usually adds meaning or adds perception or adds sensation to those sensory stimuli. But then in most cases, we can have them separate, right? I can have the sensation part, which is the mechanical part, the physical part, and then I can have the conscious perception, which is what my brain kind of puts those pieces together in order to make meaning. An example of that is hearing. So hearing is sound vibrations detected from the environment. They pass to our tympanic membrane, the eardrum, which kind of amplifies them. They pass through the ossicles, which are the inner ear bones. And all of that is sensory, pure sensation. So they're touching on these various components in the ear, but it's not until they get to the inner ear where the hair cells can pick up those vibrations, and then that can be traveled, on the auditory nerve to the brain, and then I can add meaning, which is hearing. So there's a difference between the auditory signals entering my ear, sensing, and then my perception of those auditory signals, which is sensation, or in this case, hearing. All right, really important to think about how those differ. Let's talk about the types of senses. So there are three different groups of senses that can be detected by different types of receptors, and there can be some overlap in some of these. So the visceral senses are only detecting sensory information. They're not necessarily analyzing that or adding meaning or making sensation from that. This can be stretch receptors, for example, in the body. Think about some of our like our carotid receptors. They will detect a change in the amount of blood that's passing through those areas. And depending on how they stretch, they will simply detect the stretch, right? They're not going to add any meaning to that. That's going to happen in our brain on afferent signals that go up to areas like the medulla. Another type of sense is somatic senses. Somatic senses are going to produce sensation, but these are simple and uncomplicated mechanisms, so short loops. An example of a somatic sense would be some of your muscle reflexes, right? When you strike your biceps tendon, you're going to have a response that causes a reflex, and that is purely based upon the detection of the muscle by that muscle receptor will detect the movement, and then it will have a short, sort of uncomplicated reflex loop that causes you to have that reaction, right? That muscle jerk or contraction. So that's a somatic type of sense. And then we have more specialized senses. This can be sight, hearing, and not sight, but vision, which is the meaning that's given to sight, vision, hearing, taste, olfaction. Those are going to be more specialized senses, and they're going to have more complex detection mechanisms that again separate the physical senses from the brain regions that add meaning or sensation to those sensory inputs. And although we kind of split these up and define them differently, we really integrate this all together, right? Especially because the brain is so complex. It's going to take visual information, it's going to take muscle position information, it's going to take reflex information, special senses, and then put it all together to make sense of the world around us and then make the appropriate responses. So although we kind of think about them individually, the brain does not really and blurs them all together in terms of making the best decisions to really protect us, which is what the brain does best. Let's look at how these sensations work. So we have a stimulus, which is a change in the environment that occurs. We have a structure that is on a sensory receptor that's going to pick up that change, which is the stimulus. And then that change will be converted into some type of electrical signal. And so sensing is the change. Sensing is the mechanical or receptor activity that detects the change. The neurons that are going to take that change to the brain is where we can integrate those changes and then make sensation or make conscious perception from those changes. Again, that example that I gave with hearing is a perfect example. The electrical signals that are picked up by the sound waves taken to the brain will then add meaning to whatever sounds we're hearing in the environment, right? We also want to think about adaptation. And adaptation is a decrease in the signal strength because of a prolonged stimulation, which is true. And I'm sure we've all experienced this in our day-to-day lives. You know, the more that you are exposed to a stimulus, are exposed to a sensory change, the weaker it gets over time, right? If you're in a room that's full of loud sounds, after a while, that's going to become quiet to you. It's going to become your normal. It's going to become your steady state. And then any new signals or new changes are going to sound sort of proportionately louder because that signal strength fades under constant stimulation. And this concept is referred to as adaptation. Okay. And what this does is that prepares your mind, prepares your receptors to be able to detect new changes. So this is why if you're in that room with that same sound, right? Let's say it's a really loud sound, but you detect anything different, that will become a louder or more of a sharp signal because your brain is now blurred out the prolonged stimulus so that it can be adapted to pick up any new signals and be in a position to adapt to those as well. Now, some sensors are going to adapt more quickly than others. Things like touch and smell will adapt pretty quickly. If you touch an area, your fingers will pretty much, your touch receptors will pretty much get used to it. And then you'll be able to detect any new changes in the environment. And same thing with smell. However, pain and proprioception adapt much more slowly. So you're going to have to be in pain for a very long time to get comfortable in that pain. Same thing for touch and proprioception. Okay. All right, let's talk about vision. So vision is the ability to detect light coming into the eyes, and then the ability to integrate that into your consciousness, again, adding conscious perception, and then making visual images in your brain from those sensory signals. The eyes are going to be the organs of vision, and they have specific receptors on them called photoreceptors, specifically rods and cones, that is, that allow us to detect these light signals, right? Vision is really just light that's coming into the eyes, and then those can be projected onto certain visual pathways. These visual pathways include the optic nerve, the optic chiasm that go all the way back to the occipital lobe, and that is where we can add that perception, which creates our vision. So if you really think about it, our eyes are not very different from a camera. Our eyes are purely mechanical. All they do is take a picture. In fact, they take thousands of tiny pictures in time, and then those get projected by way of neuronal signals to the parts of our brain that can add meaning to those very small, images, right? Let's begin to look at some of the eye structures and talk about their related meaning. So we're going to start by the outside of the eye by looking at the outer parts of the eye, then we're going to work our way into the eyeball itself. And we want to think first about the lacrimal apparatus. The lacrimal apparatus is the parts of the eye that control tear production, the self-cleaning mechanism of the eye, the lacrimal gland itself, which makes the tears, and then how those tears wash across the surface of the eye, and then are reabsorbed into the nasolacrimal structures. So that is the lacrimal apparatus, and we'll talk about each of the components and kind of what it does in that overall process. First off, we want to think about it as being involved in tear production, starting with the gland. So the lacrimal gland is the outer gland that we see here. It's in the upper outer quadrant of the eye. It has two parts, the orbital part that is out in the orbit or the bony socket of the eye, and then the palpable part that is closer to the levator palpebrae, which is one of the eyelid muscles. All right, it's deep to the levator palpebrae. Now, the lacrimal gland is in charge of making tears. Those tears wash across the surface of the eye from the lateral surface to the medial surface, and then they are reabsorbed by the medial lacrimal structures, and then they travel into the lacrimal duct. We want to think about how they get reabsorbed. So as they wash across the surface of the eye, they are picking up dirt, dust, debris, any discarded eye follicle, lashes or eye brow hairs that may have gotten into your eye accidentally. Those will be picked up in that fluid, and then they'll be absorbed into the anterior eye, specifically the superior and inferior puncta, right? The superior and inferior openings that allow those tears to be absorbed. So we can see those puncta here, which are these openings in the eye itself, the superior puncta and the inferior puncta, which can absorb that fluid. From there, they're going to go to the inferior lacrimal canaliculi. There's a superior and inferior. And these are basically channels. The word canaliculi means a tunnel or channel that leads to another area. And so they can go superior or inferior, and then they drain into the lacrimal sac. From the lacrimal sac, they drain down by the nasolacrimal duct into the nasal cavity where they can be absorbed. This is why when you get really teary-eyed or you have allergic reactions, you typically get sort of snuffly as those lacrimal secretions are absorbed into the nasal cavity and then eventually ingested, right? So to go over that one more time, we have the lacrimal gland, the outer orbital part, the inner palpable part. We have the lacrimal puncta, superior puncta, inferior puncta. These lead to the superior and inferior canaliculi, which are these draining sort of channels here. And then that leads to the lacrimal sac, and then down to the nasolacrimal duct into the nasal cavity. Okay. Let's look at the eye more specifically, again, working our way from the outside in. We're going to start with the extrinsic eye muscles and then talk about the inner eye structures. So there are 6 skeletal muscles that are located on the outer surface of the eyeball itself. These are important in helping the eye move, as well as protecting the eye, right? Protecting the eye, allowing it to rotate internally, externally, and helping with the self-cleaning motions of the eye as well. So there's the superior rectus. And before we dig into these, the names of these muscles will help to tell you their orientation. Are they straight? Are they oblique, right? Rectus meaning straight, oblique meaning angled, or sort of, at an angle as opposed to being straight. So the superior rectus, which is on top of the eyeball, this view here is a superior view looking down. So superior rectus is going to be right underneath levator palpebrae. Levator palpebrae is what moves the eyelid, and then superior rectus is what moves the eye up and down. Then we have inferior rectus. You have to look underneath the eye to see inferior rectus. Similar orientation in that it's moving straight back and forth, but it's on the inferior surface of the eye. It helps the eye to move down as well in conjunction with superior rectus. Then we have the horizontal muscles, the lateral and medial rectus. Lateral rectus moves the eye laterally, outward. Medial rectus moves the eye medially or inward. Okay, so those are the 4 recti muscles: superior, inferior, lateral, medial. Now we have the 2 obliques, and the oblique muscles are going to help the eye to move at an angle. So think about looking into the corners of the room. If you're looking into the upper corners, you're going to need to move your eye up, but also lateral. And that is where the inferior oblique comes in. So the opposite muscle is going to help your eye look in that direction. Okay. Same thing with the superior oblique. Superior oblique helps your eyes to look in the lower corners of the room, down, but also lateral. And if you look at the location of these muscles, that'll help you as well. So the inferior oblique, which is at the bottom of the eye, but it's also running sort of at an angle. It's attached to the orbit. And so when it contracts, it helps to turn the eye up and out. So that helps you look in the upper lateral corners of the room. And then superior oblique is going to be on top of the eyeball, does the same thing. Well, it takes the eye downward, and that helps you look down and lateral as well. Now, apart from these muscles, the rest of the orbit is really occupied by the extraocular fat that helps to pad the eye, cushion the eye, and just help to occupy the bony surfaces inside the orbit where the skull bones are coming together to form the orbit. Let's look at the gross anatomy of the eye itself. So now we're getting into the eyeball, the inner eye structures, and kind of how they're involved in vision. So we want to think about the colored part of the eye. This is one of the probably the most notable parts of someone's eye. When you look at their eye, the first thing you see is what color they are, right? This is the iris. So the iris is made up of fibrous tissue as well as smooth muscle that helps to change sometimes the shape of the pupil itself. But the iris is where we have the pigmented region of the eye. So if someone has blue eyes, green eyes, black eyes, brown eyes, that will typically be notable at the iris. Then there's the pupil, which is directly inside or being subscribed by the iris. The pupil is really an open area that helps to dilate and constrict, and this can alter how much light is being entered into the eye itself. Okay. So the pupil is really an open, dark space, and this can be a good clinical tool. We can look at someone's pupils to determine their pupillary reflexes for one. This tells us how the optic nerve is working. When you go to the doctor, they shine that light into your eye, and they determine how quickly and how consensual, right, are both pupils constricting, how quickly and how consensual your pupillary reflexes are. So that can be one helpful use of the pupil. It's really to tell us a lot about how the optic nerve is working and how those reflexes are working. It can also tell us about someone's alertness, their awareness, right? When someone's passed out, we first look at their pupil to determine what kind of brain activity they have going on, again, with those reflexes, as well as just give us indicators about just their overall state, right? So those are two very helpful, very common parts of the eye that can tell us a lot just by looking at them, the iris and the pupil. Let's move into the layers of the eyeball. What is the eye really made of? So we want to think about the fibrous layer being the cornea and the sclera. This is the outermost portion of the eye. So when you think about the sclera, which is this white region of the eye, so this outer white region that makes the eyeball white, that's going to be the outermost fibrous layer of the eye. It shows up as pink in this image, but it's really white, or it should be white if someone is in, good health, good state. So this outer sclera is actually continuous with the cornea. Now, although it's continuous with the cornea is transparent and the sclera is not. So here's the cornea covering the anterior portion of the eye. And it's really important for the cornea to be transparent because that is what lets light in through the lens and eventually back on, onto the retina, etc. So the outer fibrous parts of the eye are going to be the sclera and then the transparent cornea. The second layer is going to be the vascular layer. This is called the choroid, and this has the ciliary body and the iris. So the choroid is much more of a network layer. So it has all of the capillaries, the arteries, veins, And then it has small sensory nerves that are in this um, second layer in between the sclera and then the retina, which is the innermost layer. The choroid is also continuous with the ciliary body. Here's the ciliary body, and then eventually the iris, right? Which is the colored part of the eye. And both the ciliary body and the iris are smooth muscle. So again, these can contract and they can relax or constrict and open up, and that can change the shape of the pupil or the size of the pupil. So when the iris constricts and relaxes, it changes the size of the pupil. When the ciliary body does the same thing, it actually changes the shape of the lens. So here's the lens. Notice that it's attached to the ciliary body. And we'll talk about that attachment, which is the suspensory ligaments. But this attachment is what changes how concave or convex the lens is, and that can change where light or how light is projected onto the retina. Now, the innermost layer is the retina. This is the yellow layer here. This is a sensory layer, a nervous layer that is continuous with the optic nerve posteriorly, which is cranial nerve 2, as well as containing the fovea. And the fovea is kind of that central region that we'll talk about that's involved in the focusing area of vision, of light. All right. So those are some of the layers of the eye: the outer fibrous layer, the second vascular layer, and then the inner sensory layer. Now, when you think about the white of the eye, we've talked about this already. This is really important. It has the anterior region that is the cornea, that is transparent. I think we've discussed and described this. I want to talk a little bit about how sensitive the cornea is in terms of things like cataracts, right? The cornea should remain clear and transparent. And so this is why we need to continually clean the eye. And this is where we can have these dense deposits that can obscure vision. And these are called cataracts, right? These opacities that make the cornea more opaque than transparent. We also need to think about the uvea. Now, the uvea is the collective term for the choroid layer. The uvea is comprised of the choroid, the ciliary body, and the iris. All three of these structures together makes the uvea and are continuous as the vascular layer or the choroid in between the sclera and then the retina. And then we want to think about the just the overall structure of how the choroid and ciliary body come together, as well as the changing of the pupil. So here is the ciliary body, again, connected to the choroid layer, which is that vascular layer. And then here is the iris. All three of these structures make up the uvea. And then the ciliary body is what encircles the lens. So here is the ciliary body. And so when it changes its shape by way of the fibers that attach to the lens, this is what changes the shape of the lens as we described. Okay. And then we look at the iris earlier. Here's the iris more closely, encircling the pupil, but then also the lens, helping to change, how much of that lens is exposed and the shape of that lens. So the iris is really controlling the shape of the pupil. The choroid, or excuse me, the ciliary body is changing the shape of the lens. Okay. And then thirdly, the retina. Now, the retina is going to be one of the most important parts of the eye as far as vision is concerned. So there are 10 layers. 3 of these layers are neurons, essentially, that are picking up the neuronal signals, picking up light that's being projected through the eye and focused on to the retina, and then taking those by way of the photoreceptors, the rods and cones, and then transferring that onto the optic nerve that can go to the part of the brain that can add meaning or perception. So the axons that are gathering together to form the optic nerve, these are what pick up the signals from the retina and then transmit that signal towards the parts of the brain that can add the sensation, right? Add perception. And then we want to think about how the lens operates. The lens is what helps to direct the light, excuse me, focus the light onto the fovea centralis. Now, the fovea centralis is where we're trying to get that light focused to. So the lens will change its shape, either to be more convex or concave, to focus the light directly onto this spot, which is the fovea centralis, right? That's how we can make those images and then send those signals to the brain. Now we want to think about how vision can change, how we can go from having sharp, acute vision, to having more blurry vision. So where the optic nerve is actually piercing the eye, it's going to form the optic disc, and this is called your blind spot. So the optic disc is where the nerve actually enters. And so that's going to be a spot where there aren't as many, or there aren't any, there isn't a dense population of photoreceptors that can actually perceive that spot on the eye because it's where the eye is being pierced by the optic nerve. Now, in terms of where we're going to have the sharpest vision, that's going to be called the macula. And the macula is just on the outskirts of the fovea centralis. That's where we're going to have the most acute, the most direct vision. And then the center of the macula is going to be that sort of area that we're focusing the light on, which is fovea centralis. So looking at these two spots, we can see the difference between no vision, right? There's going to be the blind spot at the optic disc, blurry vision, which is sort of in between the optic disc and the macula being the sharpest, most acute vision, and then fovea centralis being kind of where light is being centered to create those images. We also want to think more, about the lens. The lens is what helps us change the shape to determine where that light gets focused on. So looking at the shape of the lens, and a lot of people confuse the cornea with the lens, right? They're both transparent. They both help to let light in, but they both have a different location and different function. The cornea is the outer transparent layer involved in protecting the eye, but also transparent to let light in. The lens is involved in focusing light onto the fovea centralis. So the lens can be flattened or can be kind of bulky and convex in order to direct that light exactly onto the retina, as specifically onto the fovea centralis. We also want to think about the two spaces that are created by the eye structures, the anterior chamber being everything anterior to the iris and the pupil, but posterior to the cornea. So this space here is called the anterior chamber. And again, it's bounded by the cornea anteriorly and the iris and pupil posteriorly. Now, behind the iris itself, we have the posterior chamber. And the posterior chamber is a much smaller space between the iris and pupil and then anterior to the lens. So this tiny space here between the iris and pupil, but anterior to the lens, is called the posterior chamber. All right, so the iris and pupil can divide the eye itself into two smaller spaces. Okay, let's talk about the vitreous body. And the vitreous body is really that clear, jelly, liquid-like substance that fills the eye, keeps the eye patent, and it's really in the posterior portion of the eye. All of this region behind the lens, lining the retinal layer is where we have the vitreous body. So this is really fascinating. If you were to take the eye of a cadaver, for example, usually when we remove that, you'll see fluid kind of leak out of that eyeball as we dissect, see some of the inner structures. So that is the vitreous body. That fluid is what keeps the eye open and prevent it from sort of collapsing or indenting on itself. It keeps the pressure inside to make sure that the eye stays open and patent. And not open in the sense of the eyeball or the eyelid, but open in the sense of the ball itself, keeping that open and sort of inflated. When you look at the substance itself, the vitreous body, it's mostly water, but there's going to be other electrolytes, other, um, sort of gelatinous substances that help to create a jelly-like consistency to this fluid. And that is what helps to maintain the shape of the eye, the spherical shape of the eyeball.